Hi everyone, we're done with that hard renal chapter and now we're going back a few chapters and looking at the endocrine system. We're going to start out with endocrine regulation, which is chapter 25 in our textbook. And we're going to go a little bit easier, and I think this will be an easier area of endeavor for you. I feel like we have a few people who are dropping off and we need to wait for some people to catch up to the rest of the class. So this seems like a good spot for us to slow down the pace a bit. Okay, let's just go over our learning objectives for this chapter. I want you to pay mostly attention to the hormones in the endocrine system as part of the overall system of regulating our body functions. And here are the objectives for the chapter that the book lists, which is for you to compare the endocrine structure and function with the nervous structure and function, to identify different ways to classify your hormones, and to differentiate between the mechanisms of action of steroid and non-steroid hormones. That's usually pretty significant, and you can get very deep in the weeds on doing that. And we won't get that deep. Describe the endocrine reflexes and what upregulation and downregulation mean. Discuss the chemical nature, classification, and mechanism of action of eicosanoids. And eicosanoids is the group of substances that are the prostaglandins, the thromboxanes, and the leukotrienes. And the names are just scary enough for everybody. But we've seen them a little bit before. We saw them in the immune system. Discuss the mechanisms of hypersecretion and hyposecretion. Okay, so your endocrine system and your nervous system work in coordination with each other to help maintain the internal environment of the body or to achieve homeostasis. This is why we often refer to the whole system as the neuroendocrine system. They both function to help cells communicate with each other, to integrate information, and to control or regulate the whole entire body. Neurotransmitters and hormones serve all these same functions through chemical signals that are acting on target receptors. The neurotransmitters will act in the nervous system at a short distance across a synapse. Remember, the presynaptic neuron would release a neurotransmitter, for example, 5-HT, that will act on the postsynaptic nerve cell. Hormones instead move through the bloodstream, and they can act either locally or at a distance. The effects of hormones will work more slowly but last way longer than those of the neurotransmitters. Unlike the exocrine glands, where we had to learn all those different duct formations, the endocrine glands are ductless glands, and they are composed of glandular epithelium. The secreting cells will send hormones into the bloodstream to signal specific target cells throughout the body. And they do this through a process called signal transduction. Signal transduction occurs when the binding of signaling molecules, such as a hormone or some other ligand, will bind to the receptors on the outside of the cell. Then that receptor itself is going to undergo a conformational change that will trigger a cascade of events inside of the cell. And usually this travels to the nucleus of the cell, you'll find out. The endocrine signals regulate most cells in the body versus nervous systems, which will control basically only the muscle cells and the gland cells. Here you have a nice picture that compares the effects of the endocrine versus the nervous signals. You can see in slide A, the endocrine glands are going to secrete hormones that can travel through the blood and can travel for some distance. And they will then react on a receptor on a target cell. Whereas the neurons, the presynaptic neuron, will release neurotransmitter from the button on the presynaptic neuron. It will cross the synaptic cleft and then act on receptors in the postsynaptic cell. So it travels only a short distance. Here we look at some of the major endocrine glands in the body. You have your hypothalamus up in the brain, and the pituitary, remember, that hangs down off of the brain and sits there in the sella turcica, which protects that very important pituitary gland. You'll have an anterior pituitary gland and a posterior pituitary gland. The pineal gland, which is so named because it resembles a pine nut or a large kernel of corn type thing. It's a member of two body systems because it acts as part of the nervous system and part of the endocrine system. Very importantly, it releases melatonin, which is used to regulate functions throughout the body. Remember that melatonin levels rise and fall in a cycle related to changing levels of sunlight throughout the day. In other words, the pineal gland will be important for the biological clock. In the neck area, you see you have your important thyroid gland, which will serve a lot of our metabolic functions in the body. And enmeshed in the thyroid tissue are the parathyroid glands, which are going to be really important for calcium regulation. We have the thymus gland, which is in the mediastinum, and we talked about this already for importance in our immune system. Located on top of the kidneys, we'll have our adrenal glands, also known as suprarenal glands or above the kidney. And these are important for producing some of our adrenal hormones, such as norepinephrine and epinephrine. As well, we'll have the hormones of the adrenal cortex, the mineralocorticoids and the glucocorticoids, some of which we've already learned. We previously talked about the exocrine pancreas when we were in the digestive system, and so this time we'll focus on the endocrine pancreas and the production of our endocrine cells. So our pancreatic islet cells, we'll have our alpha cells, which secretes glucagon, our beta cells that secrete insulin, the delta cells that secrete somatostatin, but we'll go through these more in the next chapter when we get to chapter 26. And then, of course, you have your gonads, the testes in the male and the ovaries in the female, and the male and female sex hormones that we'll discuss also further in chapter 26. Hormone molecules can be classified in lots of different ways. For example, you can classify them by their general function. You can identify them as tropic hormones, hormones that are going to help in development and growth, sex hormones that will target reproductive tissue, anabolic hormones that will stimulate anabolism in target cells, catabolic hormones that stimulate catabolism, and many other functional groups. And then another useful way to classify hormones is by their chemical structure, which is always scary for students, right? This method is widely used, so we'll go through this a little bit. So the basic two chemical structures that we divide the world of hormones into are the steroid versus the non-steroid hormones. And your steroid hormones are all derived from cholesterol, and you'll see they have a common nucleus, which is a 4-ring structure that all is a derivative of the cholesterol molecule, and they are lipid soluble, meaning they move well through cell membranes but don't move so well in aqueous solutions such as blood or plasma. Your non-steroid hormones are primarily synthesized from amino acids, and depending on how large these hormones are, we can classify them as protein hormones, or if they're smaller, as peptide hormones. You even have some that are just simple amino acid hormones. And as we stated, the tropic hormones are hormones that target other endocrine glands and stimulate their growth and secretion, such as adrenocorticotropic hormone, ACTH. We've seen this a little bit before, and we know that it will stimulate the release of cortisol, for instance, from the adrenal gland. Sex hormones, these will target the reproductive tissues, and we know these as estrogen and testosterone primarily. Anabolic hormones, they're going to stimulate anabolism, or the growth and development of tissue in target cells. And you know that muscle bodybuilders often abuse anabolic hormones to build their muscles up. And it has long-term consequences for them that are also very bad. The steroid hormones are molecules that are manufactured by the endocrine cells from cholesterol. Okay? And the non-steroid hormones that we talked about are primarily made from amino acids. They are not fat soluble, and so they cannot diffuse across the plasma membrane of target cells, which the steroid hormones can do. And here again is a nice chart for you to look at of all the different classifications of these hormones into the world of steroid hormones. And you can see some that are very familiar to us over here on the left-hand side: cortisol, aldosterone, the estrogens, progesterone, testosterone, and the non-steroid hormones, which is the bulk of them. Okay, so all these protein hormones, such as growth hormone, prolactin, parathyroid hormone, calcitonin, insulin, glucagon, many familiar players for us. We have these glycoprotein hormones that have a carbohydrate group stuck onto the proteins, and these are follicle stimulating hormone, luteinizing hormone, thyroid stimulating hormone, human chorionic gonadotropin, HCG, that's what's measured in your pregnancy tests. Okay, so you can look at all these. Okay, so our steroid hormones are all synthesized from cholesterol. So they all have a 4-ring steroid nucleus at their core that looks like cholesterol, and we'll see a picture of that in the next slide. Steroid hormones are lipid soluble, which means they can easily pass through all of our phospholipid plasma membranes and get inside of cells. And they can then flow through the cell and act on things, say, at the nucleus. Examples of these are cortisol, aldosterone, estrogen, progesterone, and testosterone. Now the steroid hormones, remember, also, because they're lipid soluble and can pass through the membranes, they also don't travel around very well through the bloodstream, which is aqueous. And so we'll see that they will need a hormone carrier. Just like when we talked about cholesterol, remember we had to wrap cholesterol around in a little protein coat so it could be transported through the bloodstream. Same thing here. Our non-steroid hormones are synthesized primarily from amino acids. And as I said, based on how large they are, we can classify them as either a protein hormone or a polypeptide hormone, or possibly even just an amino acid derivative hormone. The glycoprotein hormones will have a little bit of carbohydrate attached on them, and usually onto some sort of side chain. And so we saw a group of those on the previous slide. And then there's just simple peptide hormones. And as promised, you can see in this picture how cholesterol at the top forms the derivative compounds below it. Cholesterol and all these steroid compounds have the same 4-ring steroid nucleus at their core. These are all hydrophobic and therefore lipid soluble. Here are the metabolic pathways used by endocrine cells to convert cholesterol into steroid hormones. Examples of important steroid hormones include cortisol, which you see at the bottom of the second column, aldosterone, estrogen, progesterone, and testosterone. Here we see the structure of various peptide, polypeptide, and amino acid derivative hormones. Notably at the top in letter A, you can see the structure of insulin, which has 2 chains, a smaller alpha chain and a larger beta chain that have 2 disulfide linkages linking the two chains together, and there's another disulfide link on the alpha chain itself. Oxytocin, shown in B, is a pretty small peptide hormone. And then you can see in C that T4, thyroxine, is an even smaller amino acid derivative hormone, which is bound to iodine. Hence the importance of iodine in our diet. Amino acid derivative hormones are water-soluble molecules that are derived from amino acids. Most are derived from tyrosine and tryptophan. They are stored in endocrine cells until they're needed. We will further see how they act by binding to protein receptors that are on the outside surface of a cell versus the steroid that act on the inside of the cell. They'll bind to that outside receptor, which will alert a second messenger molecule inside of the cell that will act things like enzymes or other cellular proteins that could influence the expression of genes. Amino acid derivatives are produced particularly by the thyroid gland. They are synthesized, as we saw in the previous slide picture, by adding iodine to tyrosine. And we really can't talk about the actions of hormones without talking about the target cells upon which they act. Hormones signal a cell by binding to specific receptors on or in the cell. This is known as the lock and key mechanism, where hormones will bind only to receptor molecules that fit them exactly. Any cell with one or more receptors for a particular hormone is said to be a target of that hormone. Cells will usually have many different types of receptors on them, and therefore every cell with all these multiple receptors are targets of many different hormones. Here is a cartoon of the lock and key model. This is showing you that a hormone acts only on cells that have the specific, the receptors that are specific to that hormone. Because the shape of that receptor determines which hormone can react with it. Similar to when we learned about enzymes have also a lock and key model. Signal transduction basically talks about how our hormones are going to send their message throughout the cell to get it to act in whatever particular manner we need to get it to act. Each different hormone receptor interaction produces different regulatory changes within the target cell. The cellular changes are usually accomplished by altering the chemical reactions within the target cell. For example, some hormone receptor interactions initiate synthesis of new proteins, while other hormone receptor interactions will trigger either the activation or inactivation of certain enzymes and will thus affect the metabolic reactions that are regulated by those enzymes. Still, some other hormone receptor interactions will regulate cells by opening or closing specific ion channels in the plasma membrane. We also have to consider the combined effects of hormones. Different hormones may work together to enhance each other's influence on a target cell. In a phenomenon called synergism, combinations of hormones together have a greater effect on a target cell than the sum of their effects would have if each were acting alone. Combined hormone actions may also exhibit permissiveness, and this occurs when a small amount of one hormone allows a second hormone to have its full effect on a target cell. In other words, the first hormone permits the full action of the second hormone. A third type of combined action of hormones is seen in antagonism. Here, this is basically the opposite of synergism, in which one hormone produces the opposite effect of another hormone. Antagonism between two hormones can be used to fine-tune the activity of target cells with great accuracy. Hormones also have some primary and secondary actions. Typically, your textbook is going to focus on the primary actions of hormones, but it's clear that hormones have usually more than one action in cells throughout the body. The example that your textbook gives you is the hormone prolactin. Its primary action is to regulate milk production and a bunch of other reproductive functions. But prolactin has approximately 300 secondary actions throughout the body, and most of the effects of the hormone will modulate the other activities of that hormone. So, for instance, prolactin used in milk production. It's also known to modulate anxiety and depressive-like behaviors, things that will also help, like a new mother have bonding with her infant and hopefully not be so anxious and not have postpartum depression, things like that kind of coordinate together. So our hormones, as we've stated, are going to have to travel throughout the body, and they do this by circulating through the blood. And as I said, just like when we talked about cholesterol, that some hormones are going to have to be attached to plasma proteins while they're carried along in the bloodstream, because these hormones are lipid soluble and hydrophobic. And they're going to have to free themselves from their carrier proteins when they want to combine with a receptor. And you will see in hormonal tests in your life that we will measure both the bound and unbound forms of some hormones as they travel throughout the body. And because blood carries hormones nearly everywhere in the body, even where there aren't any target cells, not all hormone molecules produced by the endocrine glands will actually hit a target. So there will be unused hormones. And unused hormones don't last long. They're usually quickly excreted by the kidneys, or they're broken down by other metabolic processes in the body. Steroid hormones are lipids, and thus they aren't very soluble in the blood plasma, which is an aqueous solution. Instead of traveling in the plasma as free molecules, they attach to soluble plasma proteins. These bound steroid hormones will dissociate from the carrier before approaching a target cell. Because the steroid hormones are lipid soluble, they can pass very easily into the inside of a cell. And so, of course, we find that their receptors tend to be on the insides of the cell. So once the steroid molecule has diffused into its target cell, it passes all the way through the cell and usually toward the nucleus, where it will bind to mobile receptor molecules to form a hormone receptor complex. This is the mobile receptor model. The hormone travels through the cell membrane, it travels throughout the cytosol, it diffuses into the target cell, goes to the nucleus, and binds to the receptor and forms that hormone receptor complex. The hormone receptor complex will then activate a gene sequence and make a transcript of messenger RNA. That transcript of messenger RNA will then move out of the nucleus and into the cytosol, where it will associate with ribosomes. And you recognize this as the process of starting protein synthesis. In this way, our steroid hormone triggers protein synthesis. Steroid hormones, we'll find, regulate cells by regulating their production of specific proteins. The amount of a steroid hormone that's present in the blood will determine the magnitude of a target cell's response. And this process can be quite slow because you have to wait for the full processes of transcription and then protein synthesis to occur. And so it can be days before you see the full effect of a hormone. Now, nonsteroid hormones, because they are not lipid soluble and are hydrophilic, act in a different manner. And these nonsteroid hormones typically operate according to a mechanism that was originally called the second messenger model. In this type of signal transduction, the nonsteroidal hormone molecule acts as the first messenger, and it delivers its message to a receptor that is stuck on the outside of the target cell's plasma membrane. The message is then passed on into the cell, where a second messenger triggers the appropriate cellular changes within the inside of the cell. This concept is also called the fixed membrane receptor model. The example that is used in your textbook in figure 25-9 is that of a nonsteroid hormone using a G protein coupled receptor, which is embedded in the membrane of a target cell. It binds to that G protein coupled receptor and activates it. And this causes an integral membrane protein called the G protein to bind to a nucleotide called guanosine triphosphate, or GTP. GTP will then activate another membrane protein, adenyl cyclase. Adenyl cyclase is an enzyme that promotes the removal of two phosphate groups from adenosine triphosphate. This will form cyclic AMP, cyclic adenosine monophosphate. And cyclic AMP is what becomes the second messenger within the cell. Cyclic AMP will then activate protein kinases, and protein kinases, as you've already learned, will phosphorylate a whole bunch of different things. And so all these protein kinases will be a set of enzymes that will activate a whole bunch of cascade of other enzymes within the cell, and it is this final set of specific enzymes that are activated, which will catalyze the cellular reactions that characterize the target cell's response. So the first hormone messenger binds to the membrane receptor, and this will trigger the formation of an intracellular second messenger. In this case, it was cyclic AMP, which activates a cascade of chemical reactions that produce the target cell's response. And there are a whole bunch of second messenger systems, some that use cyclic AMP, others that use IP3, inositol triphosphate, and others that use cyclic guanosine monophosphate, cyclic GMP. And these are systems that are used over and over again across different cells and across different organisms. They're just commonly used second messenger systems. And I don't want to go too deeply into the weeds on this and have you memorizing a whole bunch of biochemistry, but just kind of know that these are second messenger systems that are used by the non-steroidal hormones, and they trigger cascades of enzymes that ultimately lead to the response that in the target cell. Okay, so some other hormones will produce their effects by triggering the opening of calcium channels in target cell membranes. Here, you're going to find hormones binding to other G protein coupled receptors, and this can activate a chain of integral membrane proteins. This will then trigger the opening of calcium channels in the plasma membrane, and then calcium ions will enter the cytosol and bind to calmodulin. And so we'll find that ultimately this is the goal, the formation of this calcium calmodulin complex, which again acts as our second messenger. And here it will influence enzymes that will produce the target cell's response. And we'll take a closer look at this in the next slide. Here is when you use the calcium calmodulin system as the secondary messenger. We start with the binding of a hormone to a fixed membrane receptor, the G-coupled protein receptor, and this activates a chain of integral membrane proteins, the G protein and phosphatidyl inositol 4,5-bisphosphate, or PIP2, that will in turn trigger the opening of the calcium channels in the plasma membrane. The calcium ions that enter the cytosol when the channels open bind to the intracellular molecule called calmodulin. The calcium-calmodulin complex that's formed acts as a second messenger that will influence the enzymes that produce the target cell's response. The second messenger mechanism of signal transduction produces target cell effects that differ from the steroid hormone effects in several important ways. First, the cascade of reactions produced in the second messenger mechanism will amplify the effects of the hormone. The effects of many nonsteroid hormones are then disproportionately great when compared with the amount of hormone present. Steroid hormones produced effects that were in proportion to the amount of hormone present. As well, the second messenger mechanism operates much more quickly than the steroid mechanism. Many nonsteroid hormones produce their full effects within seconds or minutes of initial binding to the target receptors, not the hours or days that are sometimes required with steroid hormones. Now, there is an emerging view that steroid hormones can produce both slow and rapid effects on target cells. Slow effects we've seen result from stimulating the transcription of specific genes in the target cell's nucleus, taking some time as it does to produce new proteins, whereas rapid effects can result from altering signal transduction mechanisms in the plasma membrane of the target cell. Not all steroid hormones operate according to the second messenger model. A notable exception is the parathyroid hormones, T4, thyroxine, and triiodothyronine, or T3. T3 and T4 apparently enter their target cells and bind to receptors already associated with the DNA molecule within the nucleus of a target cell. Formation of a hormone receptor complex will trigger transcription of mRNA and the synthesis of new enzymes in a signal transduction system that is similar to the steroid reception. And we'll learn more about this in the next chapter, chapter 26. We'll now go on to talking about the regulation of hormone secretion. The control of hormonal secretion is usually part of a negative feedback loop. Negative feedback loops tend to reverse any deviation of the internal environment away from its stable point or the set point value. Rarely, positive feedback loops will control the secretion of a hormone, and we'll discuss that in a minute. Responses that result from the operation of feedback loops within the endocrine system are called endocrine reflexes, just as responses to nervous feedback loops or reflex arcs are called nervous reflexes. And while your textbook goes through the insulin system as an example of a negative feedback loop, I think a more clear-cut example of this is using your thyroid hormones. We'll learn the system in greater detail in the next chapter, but basically, your thyroid produces two hormones, T3 and T4. And when the levels of T3 and T4 get high enough, the hormones will feed back to stop your hypothalamus from secreting thyroid-releasing hormone and the pituitary from secreting thyroid-stimulating hormones. These are the two hormones that will stimulate your thyroid to make more T3 and T4. But if you already have enough T3 and T4, the feedback will stop them from further stimulating the thyroid gland. Without stimulation of TSH, or thyroid stimulating hormone, the thyroid gland will stop secreting its hormones. A classic negative feedback loop. But as I said, although they're uncommon, we do have some examples of positive feedback. An example of this is oxytocin. Here, the release of oxytocin will lead to more actions that will stimulate the release of even more oxytocin. So, oxytocin receptor activation will stimulate uterine contractions by increasing levels of intracellular calcium. So you know which second messenger system is being used there, right? And this is where the positive feedback will come into play. When the oxytocin is released, it will stimulate more uterine contractions, and these uterine contractions in turn will cause even more oxytocin release. And this is what will cause the increase in both the intensity and the frequency of contractions during labor that will enable the mom to carry out a vaginal delivery. Homeostatic regulation can operate at different levels of control. So we're going to focus our attention on the specific mechanisms that regulate the release of hormones from endocrine cells at different levels of control. The simplest mechanism operates when an endocrine cell is sensitive to the physiological changes produced by its target cells. For example, when your parathyroid gland is producing parathyroid hormone, or PTH, the parathyroid hormone that is released will cause its target cells to increase calcium in the bloodstream. The parathyroid cells will in turn sense that increased concentration of blood calcium and will reflexively decrease their output of parathyroid hormone. Another level of homeostatic regulation occurs when secretion by an endocrine gland is regulated by a hormone produced from another gland. For example, in the pituitary gland, specifically the anterior pituitary, will produce the thyroid stimulating hormone, TSH, that we just talked about. TSH will stimulate the thyroid gland to release its hormones, T3 and T4. The elevated levels of T3 and T4 in the bloodstream will feed back on the anterior pituitary, and the anterior pituitary will respond to releasing hormones or inhibiting hormones that are secreted also by the hypothalamus. We see this in the next slide of the hypothalamic pituitary thyroid axis. And here you can see that our central nervous system will either stimulate or inhibit our hypothalamus into releasing the thyroid releasing hormone. The thyroid releasing hormone will act on the anterior pituitary and cause it to release thyroid stimulating hormone. The thyroid stimulating hormone will then stimulate the thyroid gland to release things such as T4 or T3. And these, you can see, have either positive or negative feedback loops on both the pituitary thyrotropes and the hypothalamus. And we'll go over this a little bit more detail in chapter 26. Another mechanism that may influence the secretion of hormones by a gland is input from the nervous system. For example, secretion by the posterior pituitary is not regulated by releasing hormones, but instead by direct nervous input from the hypothalamus. Similarly, sympathetic nerve impulses that reach the medulla of the adrenal glands will trigger the secretion of epinephrine and norepinephrine. Many other glands, including the pancreas, are also influenced to some degree by nervous input. This emphasizes the close relationship of the neural and endocrine systems, such that they are known as the neuroendocrine system. And let's look at an endocrine feedback loop. This is an example of a short feedback loop. The parathyroid gland, which releases parathyroid hormone, will control the level of blood calcium. When lactation or milk production in a breastfeeding woman consumes calcium and lowers blood calcium concentration in the mom, the parathyroids will sense the change and respond by increasing their secretion of parathyroid hormone, PTH. PTH will stimulate the osteoclasts, the cells that chew up bone, to release more calcium from storage in bone tissue, among many other effects, which will then increase the mom's blood calcium concentration to the set point level. The sensitivity of a target cell to any particular hormone partly depends on how many receptors for that hormone it has. The more receptors there are, the more sensitive is the target cell. Hormone receptors, as with other cell components, are being constantly broken down by the cell and replaced with newly synthesized receptors. This mechanism will ensure that all cell parts are new and working properly, and also provides a method by which the number of receptors can be changed from time to time. If the synthesis of new receptors occurs faster than the degradation of old receptors, the target cell will have more receptors and thus be more sensitive to the hormone. This phenomenon is called upregulation because the number of receptors goes up. Conversely, we can also talk about downregulation, in which the number of receptors goes down. This would be when the rate of receptor degradation exceeds the rate of receptor synthesis. The regulation of signal transduction mechanisms and gene transcription triggered by various hormones can also play a role in adjusting the sensitivity of target cells to a particular hormone at a particular time. For example, one hormone can affect the signal transduction of another hormone, thus inhibiting or enhancing the second hormone's effects. We'll look at upregulation and downregulation in this next slide. And you see here in slide A, we have upregulation, in which we are synthesizing more receptors than what we are degrading. And in B, you see downregulation, where we are reducing the number of our cell receptors for hormones. And now for a complicated discussion of a group of hormones called Eicosanoids. Just the word is scary, and I know that when many of you see these big, long, scary scientific words, you just want to turn away or put your head in a hole and be in denial. But really, you can learn this. It isn't as complicated as it sounds at first, so let's break it down. Okay, eicosanoids are compounds that are a unique group of lipid molecules, and they serve important and widespread integrative functions in the body. They don't meet the usual definition of a hormone, and that's why we call them tissue hormones, because they act locally in the tissue. And they form three main types of compounds: the prostaglandins, that get their name from the prostate gland because that's where it was first discovered, but act everywhere else. Prostaglandins, you'll always see abbreviated as PGs, PG1, PG2, things like that. Thromboxanes and leukotrienes. These are all pretty much derivatives from the cell membrane. And Eicosa means 20, and these are 20 carbon fatty acids attached to a 5-carbon ring. Eicosanoids are made by cells throughout the body by breaking apart membrane phospholipids and using their fatty acid tails, specifically arachidonic acid, and oxidizing these into other compounds. And you'll see on the next slide where you'll see arachidonic acid taken from the cell membrane, and there will be two divergent paths. One, the cyclooxygenase pathway, and the other, the lipoxygenase pathway. In our cyclooxygenase pathway, or COX pathway, we're going to form prostaglandins and thromboxanes. And it is at this cyclooxygenase enzyme that if we inhibit it, this is what NSAIDs, or nonsteroidal anti-inflammatories, do. These prostaglandins and thromboxanes are inflammatory molecules. And so when we take our NSAIDs, we're inhibiting the action of the cyclooxygenase enzyme from making these inflammatory molecules. And so this is how your NSAIDs, such as ibuprofen, or specific NSAIDs, COX-1 inhibitors like Celebrex, act to stop inflammation in our bodies. So let's go through and look at that in the picture right now. Okay, so here you see your phospholipid membrane up at the top, a 20 carbon phospholipid. So think of this as being, we can inhibit here where we go from our phospholipids to the formation of arachidonic acid. Here we would inhibit phospholipase A2, PLA2. And if we inhibit this activity, this is what glucocorticoids such as cortisone do. And as you know, they have wide-ranging effects throughout the body if you take glucocorticoids. And so this reduces a wide-ranging inflammation. So when somebody is having an inflammatory crisis, we give them glucocorticoids. A little bit lower down in the step, at home, you can reach in your cupboard and take a non-prescription medication such as ibuprofen, which is an NSAID that will inhibit one path here from arachidonic acid forming prostaglandins or thromboxanes. NSAIDs inhibit cyclooxygenase enzyme. Okay, non-specifically, it inhibits both COX-1 and COX-2, cyclooxygenase enzyme form 1 and enzyme form 2. Okay, ibuprofen does that. Specific COX-1 inhibitors are things like the prescription medication Celebrex. And COX-1 is just made so that you have less effect on the stomach. Okay, and you're not supposed to have all the gastrointestinal side effects that NSAIDs do. And so it only inhibits the COX-1 enzyme. All right, and then you see this secondary pathway over there, lipoxygenase, which will produce leukotrienes. And we have other medications, but I don't want to clog your mind up with those, that can inhibit the lipoxygenase pathway and inhibit the formation of leukotrienes. Okay, so what are eicosanoids again? They're called tissue hormones because they're produced in the tissue and they diffuse only a short distance over to other cells within that same tissue. And they're bioactive lipid mediators that regulate a variety of physiological as well as pathological responses. And as I said, potent inflammatory mediators. Okay? And we take NSAIDs to inhibit their actions. NSAIDs are things like ibuprofen, maybe Anaprox. What is the trade name for Anaprox? Aleve. Those are examples of NSAIDs. Glucocorticoids work up higher in the path, okay? And these will inhibit PLA2 activity. So you don't even have arachidonic acid available for eicosanoid formation. Prostaglandins, far down on that pathway, are a group of lipids that are made at sites of tissue damage or infection, and they're involved in dealing with injury and illness, and they control processes such as inflammation, blood flow, formation of blood clots, and the induction of labor. Many tissues are known to secrete prostaglandins. They have diverse physiological effects. And we've classified and isolated many of them, and we're not going into that level of detail and down in the weeds for our purposes. But you want a general idea of what prostaglandins are. And then you also have your thromboxanes and leukotrienes. And as we saw on this slide, prostaglandins and thromboxanes are made by the oxidation of arachidonic acid through the cyclooxygenase pathway, while the leukotrienes are made from the lipoxygenase pathway. The thromboxanes are members of the family of lipids known as eicosanoids, and they're named for their role in clot formation, whereas the leukotrienes are a family of eicosanoid inflammatory mediators, and they're formed by leukocytes, mastocytoma cells, macrophages, and other tissues and cells in response to immunological and non-immunological stimuli. So diseases of our endocrine system are numerous, and we'll discuss some of them more when we get into chapter 26. Tumors and other abnormalities can commonly cause our glands to secrete either too much or too little of the hormones that they produce. Production of too much hormone by a diseased gland is called hypersecretion. An example of this would be hyperthyroidism. If too little hormone is produced, the condition is called hyposecretion, and hypothyroidism is a very common disorder that we see. Various endocrine disorders appear to result from hyposecretion are actually caused by a problem in the target cells. If the target cells of a particular hormone have damaged receptors or too few receptors or some sort of abnormality, they won't respond to the hormone properly. In other words, lack of target cell response could be a sign of hyposecretion, or it could be a sign of target cell insensitivity. Diabetes mellitus can result from insulin hyposecretion, which would be a type 1, or from the target cell's insensitivity to insulin, which would be type 2, the more common type that you see. Polyendocrine disorders are caused by hypersecretion and or hyposecretion of more than one hormone, and often an imbalance of one hormone will lead to an imbalance of other hormones. Okay, so mechanisms of hypersecretion, we'll discuss hyperthyroidism when we discuss the thyroid gland in the next chapter. We can have hyperpituitarism, so something involving the whole pituitary. You can have autoimmune disorders that result from hypersecretion. And we can have hyposecretion that results in particular from anabolic steroid use. So all those big, giant, muscular, bodybuilding guys who start abusing anabolic steroids, interestingly, will have hyposecretion of testosterone and gonadotropic hormones. So men who will take anabolic steroids to promote muscle growth and increase strength can then have their body have a response to this overabundance of hormones by reducing its own output of testosterone. And this can lead to sterility and other complications. So I guess how they appear is more important to them than how they function. You have an interesting section and some of the endocrine disorders on page 569 of your test book, and I'd recommend that you read through it, but we'll go through this a little bit more at the end of chapter 26. Let's get to some of our practice questions, your favorite time here. Okay, hormones can be steroids, peptides, glycoproteins, all the above, or none of the above. And of course, they can be all of the above. The steroids being the lipid-soluble ones, and the peptides and the glycoproteins being the hydrophilic hormones. The general effect of hormones is to produce regulatory changes within the target cell. True or false? And that is, of course, true. The control of hormone secretion is usually a positive feedback loop. And we know that it is false. It's usually a negative feedback loop. We have a very few instances, oxytocin being one of them, that's a positive feedback loop. Both steroid and nonsteroid hormones exert their effects by increasing the number of enzymes that catalyze cellular reactions. And this is true. Nonsteroidal anti-inflammatory drugs, or NSAIDs, are among the most commonly used anti-inflammatory and analgesic medications. Produce their anti-inflammatory properties via inhibition of prostaglandin synthesis. True or false? And we know that this is true. We just went over this. This is that cyclooxygenase or COX enzyme pathway. Autocrine signaling is a form of cell signaling in which a cell secretes chemical messengers that travel far and bind to receptors on a distal cell. True or false? And this is false. And we didn't really go over this. You have a box on page 567 in your textbook, which goes over the different types of local hormones. And your paracrine hormones are hormones that regulate activity in nearby cells within the same tissue as their source, whereas autocrine hormones are hormones that regulate activity in the secreting cell itself. Okay, so this is local control. A regular hormone that acts on receptors in a distal cell is not going to be a local hormone, such as autocrine or paracrine hormones. Signal transduction is A, the transport of nuclear proteins through nuclear pore complexes, the transmission of molecular signals from a cell's exterior to its interior. Is it C, a process by which the contents of a cell vacuole are released to the exterior through fusion of the vacuole membrane with the cell membrane? Or is it the movement of ions or molecules across the cell membrane into a region of higher concentration, assisted by enzymes and requiring energy? And signal transduction is, of course, the transmission of molecular signals from a cell's exterior to its interior. Okay? You can recognize some of the others here, like in C, a process by which the contents of a vacuole are released to the exterior through fusion of a vacuole membrane with the cell membrane is a form of exocytosis, whereas in D, you're having a secondary active transport. The immediate effect of a steroid hormone on a cell is A, the formation of cyclic AMP, B, the downregulation of cell surface receptors, C, increase of enzyme-controlled intracellular reactions, D, the transcription of RNA, or E, activation of adenyl cyclase. And the immediate effect of a steroid hormone on a cell is the transcription of RNA. The compound that is referred to as a tissue hormone is which of the following? Growth hormone, dopamine, eicosanoid, thyrotropin releasing hormone, adrenocorticotropic hormone, or thyroxine. And the tissue hormone are eicosanoids, the prostaglandins, the thromboxans, and the leukotrienes. All of the following are nonsteroid hormones except oxytocin, calcitonin, cortisol, glucagon, or insulin. And cortisol is a steroid hormone, so it's the exception here. Steroid hormones are proteins, or steroid hormones cannot diffuse through cell membranes. Do they bind to receptors in the nucleus of their target cells, act on target cells by turning on genes, or are they transported in the blood, dissolved in the plasma? Well, the only answer that makes sense are that steroid hormones bind to receptors in the nucleus of their target cells. They aren't proteins. Proteins are a whole other class of hormones that are hydrophilic. Steroid hormones do diffuse through the cell membranes, and they act on cell targets by binding to the receptors in the nucleus of their target cell. They're not transported in the blood dissolved in the plasma, right? They need their special transport boat, some sort of carrier protein. What are chemical messengers that are released by cells and transported into the bloodstream to alter the activities of specific cells in other tissues called? And that, my friend, is the definition of hormones. And we are done with chapter 25, and we'll move on to chapter 26. We have some time to get done with this. I think I set our target date of being finished as being May 8th. Okay, so good luck, and I hope you enjoy your hormone chapters. Most people usually like these.